Hello everyone, and thank you for joining us today for the Society for Experimental Biology Christmas Lectures 2022. My name's Jen and I'm one of the Society's events officers. So without further ado, we are delighted to welcome to you the fourth and final lecture of our series entitled Development of a Christmas Tradition, Structure, Ecology and Evolution of Mistletoes, which is brought to us today by Dr LuizaTeixeira-Costa. Who is a botanist originally from Brazil, where she obtained her PhD at the University of Sao Paulo in 2019. After graduating, she worked as a postdoc researcher at the Harvard University Herbaria, USA, and later as a visiting scholar at the Hans Wissenschaftskolleg, Germany. At present, she's postdoctoral fellow in the Impact Programme, a Mary Sklodowska-Curie co-fund action at the Vrije Universiteit Brussels in Belgium. Her research combines field, laboratory and collection-based approaches to analyse the structure, development, ecophysiology and evolution of parasitic flowering plants. Thank you for accepting this invitation, Dr. Luiza. I'm handing over to you now.
Thank you all for joining. Thank you, Jennifer, Anna, and the whole of SEB for the invitation. I'm very excited to be here to talk about mistletoes, which is a very seasonal topic, considering Christmas is just around the corner. So as Jennifer said, today I'm going to talk about structure, ecology, and evolution of mistletoes. And to start us off, let me see. Oh, there we go. So this talk is based on a book chapter we had published a couple of years ago entitled Morphogenesis and Evolution of Mistletoes Haustoria. And here are the details. I'm just going to quickly switch my mouse to a laser pointer. There we go. So here are the details for the publication, from which this presentation is based on. So mistletoes have deep cultural connections in many societies and across time, and they are of great medicinal value. So we have nowadays a lot of, especially cancer therapy based on mistletoes. They're present in popular culture, and they are part of Christmas traditions in many places across the globe. But besides these cultural connections and their medicinal value, these are parasitic plants. They are aerial parasitic plants, meaning that they're not rooted on the ground. They infest the stems and the branches of the host trees. Where they germinate directly. So instead of germinating in the soil like most regular plants, they germinate directly on the branches of the trees that they will grow on to parasitize. And that the germination process and seed dispersal is often associated with birds that will eat the fruit and then disperse the seeds. And I'd like to, whenever I'm talking to a broader audience, I like to bring in the remark that mistletoes and epiphytes are quite different. Well, they're both not rooted in the soil, so they're both in the canopies of their hosts. The mistletoe is a parasite. It is entirely dependent on, or almost entirely dependent on the host. So when the host dies, soon after, the mistletoe will follow suit. So here we have an image of a dead host tree with dead mistletoes on it. In the case of epiphytes, because they're not parasitic plants, even if the host or the support tree eventually dies, the epiphyte continues to live, continues to thrive, using that support tree as really just a support and not a proper host. And while we normally, well, at least in the northern hemisphere, well, we think of mistletoes associated with the Christmas tradition and the European mistletoe, which is widely dispersed across most of the northern hemisphere. Mistletoes are actually much more diverse south of the equator. So we have Latin America as the center of diversity for mistletoes, and they're present across all forested continents, meaning they're present worldwide except in the poles. And they have a wide range of host species that they can interact and infest, and that includes conifers, palm trees, other angiosperms, so both deciduous trees and non-deciduous trees, and even palm trees. So most mistletoes will go for a wide diversity of hosts. And in these maps, we can already see that we have 5 groups of mistletoes. They're all mostly in the tropics, but they have somewhat a different distribution across the globe. We have Mesodendraceae with a very limited distribution just here in the south of South America. We have the Amphirogenaceae mistletoes, which are present across most of Southeast Asia and parts of Australia. And we have also the Laurentaceae and the Viscaceae that are present worldwide almost. And in addition to this distribution diversity, mistletoes are also diverse in terms of their evolutionary origins. Instead of having a single group of mistletoes, so I just mentioned, for instance, palm trees. Palm trees are very diverse, but they represent a single evolutionary origin. The same with orchids. But mistletoes have actually 5 independent origins. They're all within the Santalales, the order Santalales, but we have 5 independent origins. And their diversity in terms of species number represents almost 35% of all parasitic plant diversity. So it is a very important group taxonomically as well for the study of parasitic flowering plants. And what is very interesting about the evolution of mistletoes is that their sister lineages are all parasitic trees and shrubs. So all the sister lineages and their ancestor, they're all parasitic plants, or mostly parasitic plants as well, but they're rooted in the ground. So the question is how mistletoes evolved from parasitic trees, how they made the jump, so to say, from being rooted in the ground to being associated with the branches and the stems of their hosts. And here we have, again, an image of a mistletoe, the most common one in the northern hemisphere, Piscum album, and a very well-known parasitic tree, Sandalum, which is known in Australia as Quandong. So to analyze this, to dig deep into this question of how mistletoes evolved, we decided to look at the common feature of all mistletoes, in reality, the common feature of all parasitic plants, the haustorium. The haustorium is the organ that promotes attachment, penetration, and vascular connection between the parasite and the host. It is a very peculiar organ. It is both root-like and shoot-like, so we call it a root-shoot hybrid. It is really, in terms of its development, molecular development, and morphology, it is a hybrid of root characteristics and stem characteristics. And here we have examples of the haustorium in mistletoes. We have here Across the presentation, by the way, the letter P stands for parasite and the letter H stands for host. Here we have the haustorium of the parasite then over the host. Here we have the penetration, as I said, so the haustorium acts on the attachment, the penetration. Here we see the parasite penetrating through the bark of the host, then finally penetrating through the wood, and finally then making the vascular connection between the vessels of the host and the vessels of the parasite. And what is remarkable about the haustoria of mistletoes is that we have a great morphological diversity here, unlike many other groups of parasitic plants. So we have the haustorium of the sister clades, which are root parasites. Here we have the host root and the haustoria, these white circumferences. Here are the haustoria of the parasitic trees. We have what we call these epicortical roots of the parasite. We have woody galls. We have an endophytic haustorium, a wood rose, and a clasping union. So the idea here is that we wanted to compare the development of these 6 different types of haustoria and then analyze that against what we know about mistletoe evolution in these 5 independent origins. So now I'm going to shift into talking a bit of each haustorium type before we get into their evolution and development, so that you know what I'm talking about when I keep mentioning epicortical roots or woody galls. To start off with the haustorium of root parasites, and it's important, this is really focused on the Santalales. The characteristics I'm going to talk about here may not be similar in other root parasites, but for the Santalales, we have multiple connections. So these these parasitic trees, as we can see here, they will form multiple connections. So here is one haustorium, here is another one, and we can see they're connected. So the same parasitic tree will make multiple connections to one host or even to multiple hosts at the same time. And these plants are usually host generalists, meaning that they will infest nearly any root that comes their path. And they have autonomous germination and haustorium initiation, meaning that they don't require signals from the host. The seed doesn't require signals from the host to germinate, nor the haustorium requires signals from the host to start penetrating and attaching to the host root as well. Then we have the epicortical roots, which also form multiple connections to the host. So here we have the host branch in the bottom, the haustorium of the host at the top. And here we see this haustorium type, the epicortical root, forming one connection, 2, 3, 4. So again, multiple connections with the host. Again, these plants are also mostly generalists. And we have also from the epicortical root, we can have the formation of new shoots and the adventitious formations of other epicortical roots. Then we have the woody galls. Unlike the other 2 haustoria type we just talked about, they form a single connection to the host. They promote a local swelling of host tissue. So here we have the host branch. And here we can see that at the region of the woody gall, it is very swollen up due to the interaction of the parasite and the host. These plants have a varied degree of host specificity. Some are very host specific. Some will again infest a wide diversity of hosts. And here we have a remarkable differentiation of the endophyte. The endophyte is the set of tissues produced by the parasite inside the body of the host. So here we have a longitudinal section of the host wood, and then we see here all the tissues marked in white are tissues of the parasite that are growing inside the body of the host. And this differentiation of endophytic tissue reaches its apex, its maximum, when we talk about the endophytic haustorium. So just to make clear, all haustoria have endophytic tissue, but in these plants, they are limited to their endophytic tissue. They don't form anything outside the host body. Well, the only thing they develop outside their host body is their fruits and their flowers and fruits. So we see here an image of two images of Arceuthobium species, which is known as the dwarf mistletoe. And all we see here from the parasite are inflorescences or fruits. Everything, the body of the parasite is entirely inside the body of the host, forming this endophytic haustorium. And these plants are usually very host specific. So, for instance, the dwarf mistletoe will only infest the genus Arceuthobium will only infest conifers, but within the genus, you have a very high degree of host specificity. Then we have the wood roses, which promote a severe alteration of the host cambium activity. So here we have, this is what we see. We have the host branch producing wood as it normally would, but then when we have the formation of these wood roses, the host provides almost like a cradle for the parasite, from which the parasite can then obtain its nutrients. And then finally, we have what we call the clasping unions, which are a production of parasitic tissue that nearly encircles the host branch. So the parasite starts to develop around the host branch, and it can, in some cases, almost encircle it. So what we did, we analyzed the morphology and the anatomy of these 6 different types of haustorium connections, and then we plotted that against the phylogeny of mistletoes. So with the focus, I should say, with the focus on the Laurentaceae, because it is the largest family of mistletoes, and the one where we actually have all the different forms, all the 6 different types of haustoria. So that's what we see here, the phylogeny of Laurentaceae, based on the work of Liu from 2018. And then we have the different colors, representing the different types of haustorium morphology. And we see first thing that epicortical roots are quite predominant, but what we noticed is that the transition from one type of haustorium to another in the phylogeny appears to follow a pattern. We normally have a transition from epicortical roots to woody galls, and then to either wood roses or clasping unions, and or to endophytic mistletoes. So I know it's a bit difficult to see in the phylogeny. It might appear quite small on your screen, but we're going to go into the details of the development of these plants. First, we then compared the root parasites with the epicortical roots, and we noticed that they have a very similar haustorium structure, and that autoparasitism, so when the parasite infests or attaches to itself, it's very common among these plants, among both root parasites and epicortical roots. And beyond their morphology, what is more what was more interesting for us in this case was that both the Haustoria formed by root parasites and the Haustoria formed in epicortical roots share a similar origin, a similar developmental origin and pattern. So we have an illustration here showing the development of the cortical roots, and here images from Pate and collaborators showing the development of the Haustorium in brood parasites. And we can see that from the penetration of the host to the proliferation of tissues and connection to the wood, the pattern, the sequence of events is very similar. And besides, as I just mentioned, in both cases, this Haustoria share a similar exogenous origin. Then when we compare epicortical roots in woody galls, we also see a pretty similar pattern of colonization of host tissues, meaning that the way the endophyte of the parasite spreads within the host body is very similar between these two types of Haustoria, as we can see here. So we have an initial penetration, the growth of the endophyte inside the host, and then finally the connection with the host wood via a very similar structure. And we can see these similarities even more strikingly when we look at the anatomy of these plants, although the morphology might look quite different, especially because woody galls form multiple connections, as we can see here. So this is the epicortical roots form multiple connections. Here we have an epicortical root, one connection, a second connection, and then we have the woody galls with a singular connection. But in both cases, even though we're comparing, morphologically very different forms of connection, anatomically, they're very similar. We have multiple sinkers. The sinker is the structure that actually connects to the wood as part of the endophyte of the Haustorium. And we have here multiple sinkers in both types of Haustorium morphology. And then, as I mentioned before, the proliferation and the specification of the endophyte that we see in the woody galls takes its maximum development when we talk about the endophytic Haustorium. So here we have almost a division of labor with the different tissues of the endophytic system. And this is very peculiar among among mistletoes. We have very few mistletoes that are endophytic, but they're very interesting because they represent this really a specialization of the endophytic tissue, which is the part of the Haustorium that will actually tap into the wood of the host. So it is, although there are few species, they're very interesting in terms of their evolution and development. And then when we add the wood roses to the comparison, they're again very similar in terms of their the colonization of host tissues when compared to the woody galls and the epicortical roots, even though the final morphology is very different. And when we talk about final morphology, here we have another step. The development of wood roses and clasping unions is very similar, as is the development of the other Haustorium types, as I just mentioned. But then in wood roses and clasping unions, the final product, as we see here, is very similar. So when we cut a cross section through the host branch of a clasping union, it is very similar to a wood rose. So it indicates a possible convergence of the morphology as well of these two Haustorium types. So what we notice is that it's a beautiful example of the morphological and anatomical development of the Haustorium, recapitulating the evolution of these Haustorium types. So the sequence of developmental events matches really close with the evolutionary sequence or the evolutionary time and the ancestral state reconstruction in the evolution of Mistletoe. So we first have an initial host penetration for all these different types of Haustoria. Then we have the proliferation of parasitic tissues at the host cambium zone, followed by the development of the sinker, which is again the structure that will actually tap into the wood of the host. And from here, this is a crucial point in the development of a mistletoe. So we can have the formation of additional haustorial flanges and sinkers, as we can see here. So here we have a first sinker. Here we have the development of an additional sinker, or we can have, in the case of root parasites and root parasitic trees and epicortical roots, an entirely additional haustoria, not only additional sinkers, but also additional haustoria. But then when we come back to this crucial moment, which is actually the proliferation of parasitic tissues inside the host cambium, at the cambium zone of the host, if instead of having the development of the sinker right away, we have a bit more proliferation of the parasitic tissues at that area, that leads to the pathway of the formation of woody galls, because that intense proliferation will then be followed by the formation of these strands of parasitic tissue in the host bark, and then the formation again of multiple sinkers. With the endophytic haustorium, that is taken up a notch, so to say. So the parasites develop the bark strands and the multiple sinkers, and then the external part of the parasite withers and dies. And then we have this reduction of body size and restriction of the vegetative body of the parasite to the endophyte. I hope you can see the video here. This is I'm gonna play it in a minute. It is a recording of a microtomography of Arthrotobium, which is Arthrotobium douglasii, which is an endophytic parasite, to show precisely, I think now I have to switch back to the mouse. There we go. So here we see the host wood on the bottom. And we see, so the host wood is just the frame, and most of what we see in the video is the tissues of the parasite. So here we have the sinker, and then up here we have the proliferation, this remarkable proliferation and tissue differentiation into bark strands and sinker into the bark of the host. But then from this intense tissue proliferation, which is a second crucial step in the development of Mistletoe haustoria, if instead of having then the formation of bark strands, we have an alteration of the cambium activity, we end up with the morphology of a wood rose or a clasping union. And then one of the open questions of our work is what then regulates the difference between a wood rose and a clasping union. Why in some cases the host, the cradle, the actual wood rose that the host forms contains all the parasitic tissue, and in some cases, the parasite will actually almost encircle the host body. And then another parallel to our analysis of development and development of the morphology of the anatomy of the haustorium and how that fits with the phylogeny and the way we understand mistletoe evolution at present, there was a very interesting publication from my colleague, Dr. Dave Watson, showing that the emergence of mistletoe lineages coincides very nicely with the divergence of early mammals in evolutionary time. So at present, we are combining developmental and the development of morphology again with the ecology of these plants to analyze. We're analyzing fruit and seed morphological development across the evolution to see if there is a connection between mistletoes, to better understand actually the connection between mistletoes, birds, and early diverging mammals. And that is the work I hope to present at the conference, the SEB conference next year. So I hope you stay tuned for a follow-up in our quest to understand how mistletoes have evolved. And with that, I thank you all for attending this lecture, and I'd be happy to take questions.
Thank you so much for that, Luisa. Yeah, I for one can't wait to hear the follow-up at the SCB annual conference. That would be really interesting. We're just going to pop a link in the chat. Anna's kindly put a link in the chat for our feedback form. If anyone's got a moment and you'd like to leave some feedback for this event, please do follow the link and do so. So yeah, as we mentioned earlier, if you'd like to ask any questions, you can do so via the chat function or by raising your hand. So we've got a question here
There's a question.
Have you ever seen mistletoe sprout and survive on soil?
So mistletoes do germinate autonomously as well, meaning that just like the root parasites in the Santalales, they do not require stimuli from the host to germinate, but so that theoretically they can germinate on the soil, but they can sprout theoretically, but they will not survive because the haustorium and mistletoes they develop from the tip of the root meristem. So once they germinate, instead of forming a regular root, they will form a haustorium right away. So they have no way of tapping into the soil for the uptake of nutrients.
Thank you, Luisa. Do we have any more questions? Just give you all a few moments if anyone would like to ask any more. I've got a question here from Anna. You mentioned autonomous initiation. How do they know how/where to grow without any signs of the host?
Well, for most part, what we used to think is that they didn't know. They would just germinate, and if they're lucky, they're attaching to a host. If they're not, they'll just die. And that is actually the case for most mistletoes. But then there's some recent research, very interesting, showing that some mistletoes are actually capable of sniffing their hosts. So this is a very interesting publication with a mistletoe that is specific, or maybe not entirely specific, but a species that prefers oak as its host. So once it germinates, it it is able to capture some volatile compounds released by the oak tree, and then it proceeds with the growth if it is in an oak tree. And then if it is not, it will not follow its developmental course. It will just wither and die sooner than would be expected. So there's a lot we actually don't know about how mistletoes can identify host preferences. So I mentioned the endophytic mistletoes, they're quite host specific. Viscum minimum, for instance, they will germinate in absolutely any host, but they will only penetrate their specific Euphorbiaceae hosts. So maybe in some cases, it's not at the germination that these plants are able to detect their preferred or their exclusive hosts. It's at some further point during their development, but some of them are capable, and we don't quite understand the evolution of host specificity yet, but it is definitely a very interesting topic.
Thank you, really interesting. We have another question from John. What kind of defenses do hosts have against mistletoes?
Very few, to be honest, and that's why mistletoes in some areas, especially areas with intense horticulture, mistletoes can be a problem. Yeah, unlike bacteria or fungi, mistletoes are also plants, right? So any defense mechanism that the host might have against another plant, it might also harm the host plant itself. We see that in some cases, the host will try and produce a tougher bark, or a bark that is different in rugosity. So it's a bit to try and make it difficult for the mistletoe to attach. In some cases, there's intensified lignification of the host bark or the host wood to try and, again, deter the mistletoe from penetrating, but that mostly doesn't work.
Thank you. We've got another attendee with their hand up. I'll just ask you to unmute if you'd like to ask your question.
Hi
That's
there.
Garbutt. Hiya.
Hi, can you hear me?
Yes.
Thank you for a lovely, interesting talk. I would like to ask, it's been described to me that mistletoe is not strictly a parasite, it's a hemiparasite, being that they photosynthesize as well. Is that, I'm sorry if you already covered this, but what is the correct term that should be used? Are they hemiparasitic or fully parasitic?
The terminology of hemiparasites and holoparasites, which would be fully parasitic, is actually full of holes. Mistletoes are all mistletoes are capable of photosynthesizing, so they do get carbon and sugars from photosynthesis. But so in this way, they could be called hemiparasites. But at the same time, they also get carbon from the host. They don't tap into the host phloem, which is the sugar pathway within any plant, but they do get carbon from the host via the xylem, which is the water pathway. So again, that pokes a hole within this dichotomy between hemiparasites and holoparasites. On the one hand, they are capable of photosynthesis, so they definitely produce their own sugars, but they also capture sugars from the host at the same time. So more recently, we've kind of moved towards calling mistletoes their own functional group with these physiological quirks.
Yeah, that's really interesting. Thank you. Because as you explained, as soon as the host dies, the mistletoe will die as well.
It depends a bit. It depends on, well, they certainly will die. The parasite will certainly die if the host does. But some mistletoes can have a prolonged life, so to say. So in some cases, the mistletoe will die a few weeks before the host. In some cases, a few months. After the host. In some cases, a few months after the host. So we don't know. Maybe in these cases, they're using the host just as a straw to draw water from the soil. But that is, we're not entirely certain what regulates this overlife that the mistletoe can have if the host dies.
Thank you.
Thank you.
We've got one more question again from John. Do mistletoes secrete hormones to drive cambium proliferation?
They certainly do. It is a very delicate dance, so to say. So the mistletoe is secreting hormones into the host, and it's at the same time capturing hormones from the host. But definitely the mistletoe, and we know that from very few species, that has been shown for very few species, but we just assume that that's what happens in all mistletoe host associations. It secretes hormones at the cambium to make the host produce more vessels at the connection. So it feeds better the parasite. But then with connections such as the wood roses and the clasping unions, in which the host forms, that cradle almost for the parasite, that is the differentiation. The mistletoe is governing host cambium proliferation at another level. For that, we don't know what other hormones they could be or other substances they could be secreting to produce that structure. But in most cases, we were confident that there is hormone liberation, definitely.
So thank you again, Luisa. That was a really, really good talk. I'm sure you will enjoyed it as much as we did. And that brings us to the end of our Christmas Lectures 2022. So thank you all for coming to those. Thank you everyone who attended today. We hope you've all enjoyed the event, and we wish you a Merry Christmas and a Happy New Year. We hope to see you in 2023.
Full of mistletoes, hopefully.
Yeah, lots of mistletoe. There is the feedback form again in the chat, so if you wouldn't mind filling it out, if you've got a spare moment, we'd really appreciate it. One last announcement is next year is the Society's centenary year, so we've got many events planned and initiatives to celebrate our 100-year anniversary. You can find out more via our centenary page, and we've got the link in the chat there for that as well if you'd like to have a look. Okay, so thank you all for coming.